everyone. Welcome to OsteoBites today. Thank you for joining us. We'll give everyone a few seconds to get settled in our virtual room. My name is Christine Iptoma, and I am mom to Osteo Angel Dillon and Director of Scientific Programs at MIB Agents. And I am very excited for OsteoBites today because we are going to be talking about factor, which, as you know, is one of my favorite topics. And I'm joined by someone who I have the privilege of working with on a regular basis. I'm so pleased to have Dr. Matteo Trucco joining us today to give us a layperson breakdown and guide to factor. Thank you so much, Dr. Trucco, for joining us on OsteoBites today. We are thrilled to have you.
Thank you for having me.
Everyone, I should just say, please feel free to submit questions during the presentation. You can use the Q&A to do that, and we will make sure we share those with Dr. Trucco, because today is really about just kind of breaking down the factor scientific panels. And so we definitely welcome your questions. So a little bit more about our special guest today. Dr. Matteo Trucco is a pediatric oncologist caring for children, teens, and young adults with sarcomas at Cleveland Clinic Children's. He also serves as clinical director. He directs the Children's Cancer Innovative Therapy Program and co-chairs the National Pediatric Cancer Foundation's Sunshine Project Consortium, where he and colleagues develop and conduct clinical trials seeking more effective and less toxic treatments for childhood cancers. He received his undergraduate degree from the University of Pennsylvania, his medical degree from Temple University School of Medicine, completed his pediatrics residency at Rainbow Babies and Children's Hospital, and his Pediatric Hematology Oncology Fellowship at Johns Hopkins and the National Cancer Institute. We are so fortunate to have him on our MIB Agents Board of Directors, and he also chairs our Scientific Advisory Board. And on top of all that, because that's a lot of hats to wear, he also co-chairs the organizing committee for the Factor Conference and moderates the MIB Agents Tumor Review Board for Osteosarcoma. I'm surprised that you actually have a free hour to spend with us today, Dr. Trucco.
Well, I might get interrupted, but we'll see.
So this episode is a little different than our normal programming, as we will be talking all about factor and what to expect from the scientific panels and breaking it down in layperson terms for our patient caregiver advocate community to help everyone get the most out of the fabulous scientific content we have lined up this year. So if you are with us today or watching this on demand, I hope that means you are either already registered for factor or considering attending. We are reaching max capacity, so if you're on the fence, I definitely recommend registering ASAP to make sure you get a spot, because we're actually going to run out of room in the conference room. Today's talk is going to focus on the general session scientific panels and explain some of the topics, terms, and buzzwords. I will put a link in the chat to an expanded glossary of terms organized by panel topic, because as much as he will try, there's no way that we'll be able to cover all those terms in 1 hour on this episode. And because we likely have more of a layperson audience listening in today, I just wanted to highlight some of the other activities that we have at Factor outside of the general session. The conference kicks off with a welcome reception for all at 5 o'clock on Thursday, June 20th. The infamous Docs versus Warriors games take place during the reception, and then we all head to dinner together. There's a dinner for our conference speakers, and then all other conference attendees are invited to meet up at House of Blues, a fun spot for a non-hosted community dinner. And then the conference general session runs 8 to 5 p.m. on both Friday, June 21st and Saturday, June 22nd. And we'll have some wellness sessions scattered throughout the day, mainly for our patients, caregivers, families. But we also have some early morning meditation and even an early morning run with Dr. Trucco for those who don't want to miss any of the scientific panels. And throughout the day, we have some really cool wellness classes like crochet, which is actually taught by an osteo warrior, sound bath, yoga nidra, flower arranging, and therapy bunnies, which we had last year, which was amazing. And also a session on how to talk to bereaved families and more. So if you need a break from the scientific stuff, there are lots of places to connect with others in our community. Friday night dinner is always special, and we upped the ante this year with an enchanted forest prom after dinner. Our junior advisory board is planning it, and it is going to be a good time. For any patients and families interested, we also have an ambassador agents training on Thursday, June 20th, before the conference starts. And then we have an in-person Healing Hearts Grief workshop for bereaved parents on Sunday, June 22nd from 9 to 12 after the conference ends. So there are lots of ways to engage and connect, and we are so looking forward to seeing you all there. So Dr. Trucco, I'm going to hand it over to you if you want to go ahead and get your slides up. And while you are getting your slides up, I have to ask you, what song will get you on the dance floor at prom?
Oh boy. I
Sorry
don't
to put you on
know.
the spot there.
Yeah, I have to put some thought into that. I'll get back to you.
Okay. Just curious if it was like BG 70s or hip
I
hop.
mean, YMCA is easy to follow. I'll
Oh,
give you
there you
that.
go. Yeah. Okay. All
Maybe
right.
the classics.
We'll put
Okay,
that on the playlist. Okay. YMCA it is. All right. Let's get started.
good. Can you guys see the slides appropriately?
Yes. Looks great.
Okay, wonderful. Thank you, Christina, for that introduction. And again, it is an honor to be involved with anything MIB, in particular the Factor Conference, which is kind of, at least for me, where it all started. And for the people listening and people attending, we don't pull punches at the Factor Conference. These are heavy scientific presentations, the same kind that we would be presenting at, the top scientific conferences. And while we do try to kind of tailor it to the audience, these are just very, complicated topics. And so we thought maybe it would be helpful to kind of go over some of these topics ahead of time, kind of just explain it. I'll do my best to explain it in layman's terms. And maybe that'll help people be a little less intimidated by the conference itself and maybe get a little bit more out of the panels. So with that, we have 9 scientific panels. Again, how Christina manages to fit them all in a couple days, I don't know. And we're trying to target kind of all the aspects of osteosarcoma that we can. There is a lot of overlap in these sessions, and that's okay, but we try to group it up in a way so that it makes sense. And quite frankly, also we want the people that are on those panels to kind of talk to each other. So there have actually been some collaborations that have come out of the fact that these people were sitting next to each other. We'll kind of go through them one by one, kind of a high level look at them, and go over some of the topics. Again, it cannot be an exhaustive list, but if any questions come up, I think Christina's paying more attention to the chat. I'm happy to answer them as we go along. So the first panel is how AI will impact clinical practice and research. So AI being artificial intelligence. And everybody's heard about ChatGPT and how AI can help generate documents and also be used in bad ways and kind of make fake videos and stuff like that. Here, the power is hopefully harnessed for good, and just using computers, ability to process massive amounts of medication, of information, and recognize patterns, and kind of see things that we aren't able to see because as smart as we might be, we just can't, uh, process trillions of pieces of information the way a computer can. So fortunately, we have some experts in the field focusing on oncology and specifically in osteosarcoma. Satith is actually here in Cleveland, and he's going to be chairing the session, even though that's osteosarcoma is not specifically his interest. And people are looking at AI in a variety of ways. One is how to kind of use it to assess the pixels on the scans that we get on patients and try to extract even more information out of those kind of white and black and shades of gray that we see. And at the same time, also kind of using almost like computer generated models of the patient and how to use this kind of digital avatar. You can almost imagine it's like yourself in a computer game and kind of getting all the medications and seeing if that can predict how the patient would respond to the same medications. So there's a variety of ways AI can be used to better assess the data that we do have, generate more data, and be predictive for us. So some of the terms, and again, I'm no artificial intelligence expert, so I'm kind of learning these things along the way. And generally speaking, all the docs in the room, they don't necessarily know everything that's going on. They're just better at, faking it. Uh, but, as far as machine learning, it's basically the computer teaching itself, looking at the data and saying, okay, all people with, whose name starts with this letter seem to have, this response to this drug. Okay, well, let's test that theory. Okay, include more patients and say, oh, no, we were wrong. It had nothing to do with it. Okay, let's look at something a little more tangible. And again, the computer is going to keep learning, and the more it looks at it, the more data it analyzes, it can make more accurate predictive models or make sense of the data more and more and more. Part of that is natural language processing. And so, if you're looking at a scan, I mean, literally the computer is viewing that as like pixels, binary elements. Is the pixel on or off? And see if they can kind of piece together more information from the pictures. Natural language processing is trying to do the same thing from our notes. And fortunately, nowadays, doctors, you don't have to worry about doctor handwriting because most of our notes are in the electronic record. So either we're dictating it or we're typing it. How can the computer extract the language out of our medical records and the notes that doctors write to make sense of it? On top of that, you can imagine the challenges of typos and stuff like that. It's no small feat, but it's a way of kind of mining this massive amount of data in all of the notes that we have in our electronic health records, and maybe we can extract some information from that again, as a single person, you can't make sense of it, but a computer just has so much more computing power to be able to churn through all that data and try to make sense of it. And digital twin, that's kind of what I had alluded to before. You can kind of, make a program where you have a completely digital person. If you think of like, SimCity or, Fortnite or any of those games, Minecraft, where you have kind of a digital representation of a person going through a world. But instead of a world, it's literally kind of testing what drugs might be doing and how their tumor might be responding to those drugs, using all the data that the machine learned to then build kind of predictive things and test a variety of scenarios uh, at a much higher speed than can be done in real life. And you can imagine one day we plug in your information in a computer and test out 20 different therapies and see which one is the right therapy for that one patient based on what we know so far. We're not there yet, but that's kind of what we're trying to aim for. So it's an exciting new world. I'm really looking forward to this session because it's a lot of stuff that I don't necessarily know a lot about. I know Cleveland Clinic has a quantum computer, so quantum apparently is better than super. So it's all things we're looking forward to and trying to figure out the best way to apply it to osteosarcoma. Any questions about that? Hopefully not, because I'm not an expert on.
Yeah, feel free everyone to just add any questions as we go to the chat. And I think it's super exciting. And that's actually going to be our first panel of the conference. And I think we'll really set the tone because all the work that's happening here, I think will really help accelerate kind of all the work that we're going to hear about in the other panels. So very excited for this panel as well.
Okay. Shifting to something that I know a little bit more about, preclinical models. So preclinical models in oncology, it's basically how can we study the cancer and the biology and the drugs and all that before we give it to patients, because you don't want to give something to a patient that's not going to work. You don't want to give something to a patient that's going to be overly toxic. Um, so, this is all the stuff that people experiment in the lab, trying to understand osteosarcoma better. So understanding its underlying biology, uh, testing different drugs against it, looking at the toxicity of the drugs, and trying to see if we can, personalize therapy to the specific tumors. All of that needs a model to be testing these things on. Each model is, by definition, not perfect, um, but they each have their advantages, and we have various people trying different kinds. So again, a slightly bigger panel looking at this in all different kind of ways. One of it is trying to understand not just the cancer itself, but the environment around the cancer. Um, some of these speakers are kind of developing novel models, be it using bone marrow or collagen to construct kind of three-dimensional models of these cancers. And we also have the last speaker, Josh. He's validating all these preclinical models to try to see which one is which ones are actually kind of the better representations of osteosarcoma itself, so that we should maybe bias studying, testing drugs and so forth on those models as opposed to other ones that aren't as reliable. Just going over some of the terms that you'll hear, the most basic model we have is what we call cell lines. That's where we take a tumor, break it down into single cells, and put it in a petri dish that has a special coating so that the cells stick to that petri dish and they grow. And, back when I was doing research, that's what I worked on. And it's great. You can have massive amounts of cells in an incubator, and you can test them, and you can put different drugs in the petri dish. It's just, what we've learned is that cells change when you're growing them on plastic as opposed to growing them in a living, breathing body in 3 dimensions. And so we've kind of gotten away from them. They still have value, for sure, but generally speaking, the field is realizing that there's only so much you can get from testing cell lines. Next is organoids and 3D cultures. So this is, you're still growing it in a test tube, so to speak, but you're trying to recapitulate that 3-dimensional structure. And so sometimes you have the cells kind of hanging, suspended, or you have them in a fluid or a matrix that kind of keeps them not stuck to the bottom of the dish, so that they can kind of grow in a 3D structure. And you can get some interesting information about, drug penetration, because if it's a flat surface, yes, all the drug goes there, but tumors, we know, take up space. So how do you get drug into the middle if the tumor's bigger? Still, though, these are cells, and some of these cells are from the 1970s. I mean, they've been around, and kind of you keep them going in the labs. So again, how realistic are those representing, our current patients? Patient-derived xenograft kind of gets at that, where we are taking a patient's tumor directly out of the patient and putting it into a mouse so that it's growing in a living thing, animal. So this is perhaps as representative of the patient's own tumor. That's good. I remember when I was a lot younger, sitting in the OR with a little tube, waiting to get that piece of tumor, and then running to the lab and implanting it in a mouse. To do that, though, the mouse has to be a special kind of mouse. It can't have a normal immune system, or it would attack the tumor right away, because it's human. It's not mouse tumor. So these are mice that were designed specifically to have suppressed immune systems so that the tumor is allowed to grow. but like three-dimensional structures still form, metastases still form because the tumor spreads throughout the mouse's body. Blood vessels feed into the tumor. You can test drugs by administering it to the mouse, either through an injection or through their mouth. Um, so there's those aspects. The mice models are really good. The downside is that immune system is not intact. So any immunotherapy, you're not really going to be able to test it accurately. And what we're learning is that the immune system plays more and more of a role in, how these tumors are actually procreating and spreading and actually are resistant to some therapies. So again, definitely some advantages, some disadvantages, and then you need the whole facilities to keep the mice alive and veterinary support and all that. Couple ways to kind of maintain the mouse's immune system, but still have tumors are what are called genetically engineered mouse models, or GEMs. Here, we mess with the genetics of the mouse to put them at a higher risk for developing tumors. And there's a couple different models out there. These are great because the cancer arises spontaneously. It arises in a mouse with a normal immune system, and you can, learn a lot from it. It's just, you know, is if you're kind of tricking the mouse's biology into generating osteosarcomas, how really accurate are those? Are those real osteosarcomas? And are you more kind of creating a model like a Leaf-Raumani or another syndrome that kind of predisposes you to developing tumors, as opposed to someone who is otherwise perfectly healthy, doesn't have any predisposition, but somehow an osteosarcoma forms? So again, definitely some advantages, but how well it represents osteosarcoma that arises and in someone spontaneously, unsure. Syngeneic mouse models, these similarly allow you to grow a tumor that pretty closely resembles an osteosarcoma in a living mouse with an intact immune system. The challenge here is that it is what looks like an osteosarcoma that grew or developed in a mouse. It's just, you're testing mouse osteosarcoma, not human osteosarcoma. Are there parallels? Sure, but they're not exactly the same. By testing different models, different things in different models, and taking advantage of the advantages of each of them, we can create or test some new therapies in the lab and learn a lot. So not all of these are, just because they're flawed doesn't mean they're useless. And again, the speakers are talking about even new models that they're developing to try to overcome some of the limitations. Any questions about those? Yes, we do handle the mice with our hands. We have gloves, but oh, and I'll also mention, it's not on the slide, but osteosarcoma has the advantage that osteosarcomas occur in pet dogs with a very high rate. In fact, there's more pet dogs with osteosarcoma than human beings that get osteosarcoma in the United States. Upwards of 10,000 a year in dogs versus more like 600 a year in humans. So you'll see there's a lot of veterinarians giving talks at this conference. And again, it's because we realize that we can learn a lot about osteosarcoma. Again, dog osteosarcoma, not human osteosarcoma, but there's a lot of parallels. And these are dogs that it happens spontaneously. They're not genetically engineered to develop those tumors. They're just your run-of-the-mill dog, but they develop osteosarcomas, and so we can learn a lot from them and even test some therapies in them in clinical trials in dogs that then hopefully can be translated into human beings.
Thanks for that nice roundup, Dr. Trucco. I think because those are terms that come up a lot, and I think this actually will also be early on in the conference and really foundational because likely in some of the subsequent talks, you'll hear some of these terms come up since all the research uses some type of one of these preclinical models. So I think it'll be really interesting to have this discussion earlier in the conference and kind of learn about those pros and cons that you went over, Dr. Trucco. And there are some exciting, like I think Dr. Freeman is actually working on organ on a chip, which is an exciting new technology. So again, all these kind of advances are kind of enabling some really interesting model types that I think can be produced, more quickly and also are less expensive. So yet to be seen, I guess, though, how they compare to some of the more traditional preclinical models, but
Yeah.
should be a good panel.
And again, kind of bridging it, I'm sure there's going to start talking about AI being used to assess some of these preclinical models. So, yeah, they all kind of work together. Shifting to targeting metastatic drivers. So, you know, I've heard this said a lot. It's not the osteosarcoma that the primary tumor that gets you. It's the metastases. It's when it spreads to the lungs that's problematic. And so there's a big focus on how can we prevent metastases? How can we, stop metastases, treat metastases? And how do we have to understand how the metastatic process happens to before we can start targeting it. So we have a whole panel looking at that. A fifth bout of osteosarcoma spreads at the time of diagnosis, and then we know a lot of patients, even if their tumor was in one spot at the beginning, and they're treated appropriately, and everything goes well, there's still a risk for the osteosarcoma to come back, and usually it's in a metastatic site, the most common site being in the lungs. And we think that's because the cells are traveling through the blood, and blood vessels from the leg to the heart get bigger and bigger and bigger, but then from the heart to the lungs, they get smaller and smaller and smaller. And so we think it might be just the physicality of the size of the blood vessels that might be what contributes to why metastases happen in the lungs more often than anywhere else. Again, a great panel from kind of all over. We have some people from veterinary medicine and people focusing on, osteosarcoma in kids directly. So a great panel looking at, again, what's driving these metastases, and then can we figure out something that'll help us treat those metastases? So, and develop new therapies for it. One of the topic that'll come up a lot is this tumor microenvironment. So, we're talking about the tumor and, as we mentioned, growing the tumor in the petri dish. Well, one of the reasons growing it in a petri dish isn't an accurate representation of what's going on in the body is that there's a whole lot of other cells there around the tumor, be it parts of the immune system, the body's defenses, just the lining of cells and the connective tissue that's around the blood vessels. All that actually we're finding out plays an important role in the development of metastases and even resistant to therapies. So getting a whole picture of what that environment looks like. Ryan Roberts, who's one of the speakers on this panel, has this beautiful kind of series of images showing how he's kind of building what's the process of developing a metastasis in the lung look like. And he kind of shows one cell labeled with, like a red dye setting up shop. But that's it. It just sits there. And then another osteosarcoma cell labeled in green meets up with the red one and then starts building things. And then they start forming kind of these structures around it to eventually lead to the metastasis. And we have to understand kind of that whole process before we can target it. Tumor heterogeneity has to do with the fact that even one tumor, not all the cells in that tumor are the same. And certainly we know there's differences between the primary tumor and the metastasis, and even probably differences between the cells within the metastasis and cells within different metastases. So it's that heterogeneity that where learning is more and more important, and to the point where maybe we're actually treating more than one cancer cell. I wouldn't say with more than one tumor, but more than one kind of cancer cell or population of cancer cells at one time. And that's uh, complicating our ability to treat this disease. Gene enhancers, that all has to do with what is being turned on and turned off in these cancer cells. We know it's dysregulated. We know proteins and signals and signals to grow and to divide are being turned on in these cancer cells. And uh, depending on what those are, that might be what's driving these metastases. And if we can find a way to like shut off one of these enhancers that's essential for metastatic spread, that could potentially be a game-changing therapy. CRISPR screening, that's a powerful new technique that's come out in recent years, where basically it lets us kind of turn on and off genes throughout the entire DNA of a cell and see kind of what's important and what happens downstream when you turn on and off these genes. Again, it's kind of like a screening of a lot of them, and then you have to kind of go to each individual gene and kind of test them a little more specifically. But you can imagine if you're going through all the DNA and there's billions of base pairs in DNA and thousands of genes that are being turned on and off, you can generate a massive amount of data with a technique like CRISPR, and that's where analyzing that will require more and more powerful computing. But hopefully with these techniques, we're getting a better understanding of the metastatic process. And once we understand it better, we can intervene and treat it more easily. That was a lot for me. I'm not sure that was a lot for other people.
I applaud your being able to explain CRISPR in layperson terms. It's
I'm,
hard.
I'm not sure if I did, but I tried. Okay. Shifting again to overcoming drug resistance. So this is a quote from actually a colleague of mine, or a paper from some colleagues of mine, Jake Scott, talking about, cancers develop resistance. I mean, we know that. You can't keep hitting them with the same thing over and over and over again. I mean, in an ideal world, and a lot of times it happens, you hit them with these three drugs, specifically more than one, because you don't want resistance to develop. And you eliminate all the cells, and that's great. But all it takes is one cell that kind of resists that and figures out how to avoid that cancer, and suddenly that population grows, and you have a whole population that's resistant to that prior chemotherapy. That's why a lot of times we don't give the patient the same treatment when they relapse, because we say your cancer has demonstrated that it doesn't necessarily isn't sensitive to that chemotherapy. There are exceptions, but so the development of resistance is certainly something that we encounter and we have to address. Again, a lot of talks on the subject, looking at everything from using genomics to kind of assess mechanisms of resistance. Looking at specific resistance to one of the common chemotherapies, methotrexate. And then some of these kind of principles related to, how does the resistance develop? Um, there's this term quiescent cells. They just sit there. They don't divide, which is weird because cancers tend to divide and grow. But they kind of ride out the chemo, and then at some point they start dividing again. And chemo kills cells that divide. If it's not dividing, it's not going to be killed. But somehow that cell remains resistant and then gives rise to new tumors. And then this concept, collateral sensitivity, is as you treat them with one drug, maybe they become sensitive to another drug. So this all falls under this evolutionary biology. It's kind of thinking, you know, we we understand certain aspects of evolution, the dodo bird, the passenger pigeon, we have dinosaurs. We know they were here, something happened to them, and they became extinct. And, evolutionary biology is looking at some of those principles that change animal populations, species populations, and try to apply some of those principles to try to make cancer extinct. Um, and so again, the dormant or quiescent cells are those cells that we suspect are sitting around doing nothing until one day, a year, 2 years, 10 years after you were in remission, they start dividing again and give rise to a relapse. Chemothripsis is also an interesting term that I'm not sure how well I can explain in layman's terms, but basically it's you have these chromosomes, which are the packaging of all the DNA in the cell. And just imagine shattering it and then watching them reassemble, and they reassemble in haphazard ways. And osteosarcoma is one of the tumors that seems to suffer from this, where there's just huge amounts of mutations in the tumor, and how to potentially kind of take advantage of that um, to treat osteosarcoma would be helpful. But it's also a way that it mutates so much that it can develop resistance, because resistance is essentially a mutation that is advantageous to the cell. And so better understanding chemothripsis is certainly important. Collateral sensitivity, as I mentioned, is this principle of I treat you with MAP chemotherapy. As I'm treating you with that, what does that make the cells sensitive to or resistant to? And as resistance to one regimen develops, does it create a sensitivity to something else? And can you kind of like predict what the cells are going to become sensitive to and kind of stay ahead of that resistance and kind of steer them more and more towards either a um, less viable population, that would be great, or a less metastatic population, or something else that will enhance our control over the cancer. And clonal evolution is this, again, this concept of how um, as the cells are dividing, whether because of chemothripsis or just mutations that happen because these cells are not normal cells, new qualities of those cancer cells are going to evolve in different populations, some that are growing faster, some that are growing slower, some that grow close to blood vessels, some that grow further away from blood vessels, will keep emerging. And so the tumor you're treating now is not necessarily the tumor that you're going to be treating in the future. That's why a lot of times we rebiopsy the tumor to send it off for genetic testing and to see if anything new emerged and potentially something that we can target. Any other thoughts on that? This is also, I think, going to be a really interesting session, especially because we're just attacking it in just all kinds of different ways, which I greatly appreciate. And it's always nice to hear Dr. Healy talk.
Fan favorite.
Hmm? We're okay on time. Shifting the key pathways for osteosarcoma treatment. Again, this has a lot of overlap with other panels, but it was a good way to kind of group together some of them. Pathways are just, something in the regular biology of the cells, dividing, growing, spreading, moving, what have you, that might be driving the cancer process or metastases. And is there some way we can better understand those drivers and in so doing intervene to stop them? Again, researchers from all over talking about it in very different ways. One looking at certain gene that's turned on, and we know that's driving the tumor, and it's particularly bad. Some other ones more kind of looking at processes like DNA damage repair. So these cells, as they're being treated with chemo, and just because of their nature as mutated cells, have a lot of DNA damage. How can we interfere with that pathway or that process to overcome some of the resistance that happens? And then looking at some of the other aspects of this, even responses to stressors or the immune system. Again, someone from Houston really wants to talk to me. Okay. Um, oncogenes are genes that, if mutated, can drive cancer. If it's a signal, a pathway that says divide, and it's stuck in the on position, that uncontrolled cell division, that is cancer. I mean, in a nutshell. So there's a lot of genes like that. MYC is one of those very uh, familiar uh, oncogenes that is turned on in many cancers and a subset of osteosarcoma. Those signaling pathways, I mean, these are just, half a dozen or so signaling pathways in the cell. And I can tell you this is an extremely simplified version of those pathways, because actually all those pathways interact, and it's more like a net, a mesh, a complete um, scribble of how these things interact with each other. And that's part of the reason why it's so hard to target any one of these, because you block one and the other one gets turned on, and then you have to block that one, and you have to kind of um, see if you can block many of them, or somehow they all funnel through this one area. You try targeting that, and maybe that's enough to catastrophically kill that osteosarcoma, because it's so dependent on that pathway. And DNA damage repair, as I mentioned, it's a process by which the cells fix their DNA, and all cells need that, um, including cancer cells. But again, there's clearly some dysregulation in that in osteosarcoma. P53 plays a role in DNA damage repair, and we know that that is mutated in the vast majority of osteosarcomas. So um, that pathway also is likely to play an important role, or we know it plays an important role. Targeting it might play an important role in developing new therapies.
I should point out that in the expanded glossary, which there's a link to in the chat, there's kind of a longer list of all the different pathways if you're interested. I think we just highlighted here the DNA damage repair because that is one that's being looked at a lot in osteosarcoma. It's a lot of alphabet soup, like even as I'm looking at the illustration here. But if you're interested, there is a longer list in the expanded glossary.
Yeah, we definitely like abbreviations in medicine, for sure. Okay. Creating an osteosarcoma atlas. So this actually came out of one of the factor conferences. We got some people sitting next to each other and talking to each other, and they're collaborating. Basically, an atlas is, can we put all this information, all this data, all these resources together in a coherent way to kind of get information such that, just like an atlas, you can open it and kind of see maps, might direct you in a certain path. Again, lots of people coming at it in different directions, and that's great. And you'll notice omics mentioned a lot, and you'll also, this is where AI might be necessary to put together and make sense of these massive amounts of data. The amount of data from one single cell is huge. And you can do analyses on multiple cells, on multiple tumors, on multiple people. And again, it just becomes a huge amount of data. But if we can kind of put it all together and harness it, and put it in a way that can be analyzed easily, we can certainly find out more and understand more about these tumors. So that's what all these terms are. Transcriptomics, I mean, you're looking at the DNA and trying to see, the DNA is in the instructions for a cell, and what parts are turned on, what parts are turned off. I'm actually going to hop over to here where RNA-seq is looking at, okay, you have the DNA, you see what's turned on and turned off. RNA-seq is looking at what signals are actually making it out of the nucleus, which is where the DNA lives, and getting outside into the cell where things are made. And RNAs that those instructions that are being relayed to the cell to make these proteins. Hopping back to the other side, proteomics, that is looking at all the proteins that are actually made in the cell, probably because of the RNA that came from the DNA. And so kind of getting a snapshot of all the proteins being made at any given point in time in a cell can be perhaps more informative than the DNA itself. The middle two terms are kind of how do you put this all together? Be it the DNA, the RNA, the proteins, also the cells. Again, we talked about the microenvironment. The physicality of where the cells stand compared to the blood vessel, compared to the immune cell, plays an important role. And it's not, we haven't fully figured it out, but that's really important. In data visualization, it's just a way of kind of putting this all into a way that we can look at it and make sense of it. And that's honestly hard. You can imagine when you get an email and it's just like words and, your eyes cross and you stop reading after a couple lines versus if you pop in a figure here or there that explains what you're looking at. Oh, okay, now I get it. And again, this figure right here kind of does just that. It looks at several of the immune cells and how they relate to the tumor cells and how the tumor cells might relate to the things around it, and signals and messages that are being sent around. So, again, this is a great initiative to try to kind of collect all this information. And I think, be it AI, be it all the omics being used, will help make sense of all this data. Going from the huge and kind of almost abstract, I have a panel on local control. And, at a certain point, you have to physically take care of the tumor, be it in, say, the bones of the leg or in the lungs. And we have really three techniques for those: surgery, radiotherapy, and interventional radiology uses techniques to kind of like burn or freeze or do something else to kill the tumor. So again, some of this is kind of looking at aspects of how we do the surgery and when we do the surgery, how does that impact survival? The planning that goes into some of these surgeries are getting more and more sophisticated. And then some talk about radiation, which again, we don't always talk about radiation because osteosarcoma is very radio resistant, but maybe we can give radiation in different ways and that'll be more efficacious. And so this multi-site SBRT and flash radiotherapy are some kind of newer techniques that are being applied to osteosarcoma with some success. And then interventional radiology, it's a way to kind of like either burn or freeze. Those are the more common techniques under image guidance to try to kind of locally kill those osteosarcoma. It doesn't kill the cells floating around, but it does address the specific tumors, which are important. Just some of the terms: allograft. Allo means different, so it's coming from someone else. Specifically here, we have banks of cadaveric bones that the person that owned them don't need them anymore. And so we can kind of say we have a patient of this height and we need this bone, and someone can go to the bank and say, yes, we do have that bone or one that matches, and that can be used. And instead of using a metal rod, sometimes that can be more functional and help with healing. Intercalary is another term that's used, and it just means kind of replacing the bone in some way. One technique, for example, is they stick a metal rod into the bone and then have part of that rod sticking out of the skin so that, and it heals nicely. There's a nice seal there, but you can actually put the prosthesis kind of into that socket that's sticking out. It just provides more of a, it's attached to the patient as opposed to, um, a prosthesis on after a patient has an amputation, kind of just it fits into a sleeve. Um, so intercalary devices can do this osteo integration is the other term for that. Dosimetry and flash RT. So dosimetry is just how much radiation you're delivering at a specific time. Flash RT is this new technique where you're delivering massive amount of radiation very quickly, and it has advantages in hopefully kind of being more toxic to the tumor and less toxic to the normal cells. And as we're getting more and more precise in how we can deliver that therapy, it helps. Imagine, you're trying to treat a tumor in a lung. Someone's breathing, lungs are moving. You can tell them to hold their breath, but good luck holding your breath for very long. And even if you're holding it, your lungs are moving somewhat. So Flash RT kind of enables us to deliver that dose very quickly, and it certainly has some advantages. And then cryoablation and radiofrequency ablation, again, those are two techniques for ablating, which is a way of eliminating, let's say, or burning or killing the tumor. One way, this is, a needle that you put into patients asleep or numbed, and you put it actually into the tumor. And then they actually put liquid nitrogen through that needle to create this ice ball. And you can see under a scan exactly how big the ice ball is and make sure it covered that whole tumor, and anything that was in that ice ball is dead. So you can completely eliminate a tumor that way pretty successfully. You also kill a bunch of normal tissue around it, so it has its limitations, but it's a technique that's pretty well tolerated. You don't have to interrupt chemotherapy or anything for it. Radiofrequency ablation kind of does the same thing, but with like high frequency electric currents, and kind of essentially burns it as opposed to freezing it, but same concept. And again, some of these can be very precise and not damage too much of the normal tissue at the same time.
I was just going to say, I'm very excited for the Flush
Mhm.
presentation because that actually came up a couple years ago at Factor. And at the time, everyone was kind of like, I don't really know what that is. So,
Right.
Dr. Frankert from Cincinnati, I think they're actually the only institution doing a trial right now, at least in pediatrics. So, very excited to have him come to talk about that. And I know for the ablations, just to do a little cross plug, our AYA podcast, Osteo, had an episode recently on their patient experiences with different types of lung local control methods, surgery, and ablations. And, a lot of them just talked about the recovery time for those ablative procedures was so much shorter. So it'll be really great to hear from Dr. Sheikh on those interventional radiology techniques.
Cool. Yep. Okay. In the sake of time, I'm going to try to speed up a little bit. Immunotherapy, it's using the body's defenses to fight the tumor. Really cool topic. Every year we kind of say, well, we talked about immunotherapy last year. I don't think we need a panel. And every year we end up with a panel because there's just a lot of interesting stuff happening. New techniques, new targets, trying to make the targets we have better. There's a lot of different approaches to this, and we're fortunate to have a lot of experts in the field focusing on osteosarcoma. Um, cytokines are the signals that the body gives to stimulate the immune system. I love this little cartoon to explain what the checkpoint inhibitors do. They're kind of these little stop and go signals here. Depending on how the T cell is stimulated, it either responds to the stimulus of go find this cell and kill it, or it doesn't. And checkpoint inhibitors block the signal that tells the T cell to not react. So by blocking that signal, the T cell is stimulated to react. The surfaceome, so we're adding ohm to everything, basically looking at everything that's on the surface of the cell. The immune system can only react to something that's on the surface of the cell. That's what kind of antibodies do. That's what the T cell receptor does. It's touching the outside of the cell and saying, what can I tell about you based on what's on the surface? And the surfaceome is taking all those signals, all those proteins, all those sugars on the outside of the cell, and seeing what we can use from that to better stimulate the immune system. Dendritic cell vaccine is where we take some cells out of the body, expose them to an antigen, which is basically, usually it's a protein, or it's, again, something that's on the surface of the cancer cell, and say, hey, this is what we want you to kill. We stimulate it. We energize those cells, and we give them back to the patient, and those cells go around, and that's the purple cell right there. They go and try to stimulate the immune system to say, hey, go eat this, or go attack these cells that express or have this protein on their surface. And that's how the dendritic cell vaccine works, is we're taking your cells, we're training them what to stimulate the immune system to respond to, injecting it back in, and they go and stimulate your immune system. Tumor infiltrating lymphocytes. So lymphocytes are, that blue cell. They're the ones that do the cell killing. And we know there are lymphocytes in tumors. They're just not doing what they're supposed to. And so what can we do to stimulate them? Is it a checkpoint inhibitor? Is it that they need more motivation by the dendritic cells? Or is there something else going on? So we're learning more about them. And then CAR T cells are basically engineered T cells, where we tell them specifically what thing on the surface we want them to recognize and go after and kill. And so again, we have various targets for CAR T cells, various generations that are stimulated and more aggressive. And again, I think we're in generation 4 of CAR T cells, so they're getting better and better. Noticing some chats. I don't know if there's any specific questions.
I'll answer the question in the Q&A. There was a question about whether or not this will be posted, and the answer is yes. So
Oh, good. Okay. Clinical trials, I mean, that's what all this is coming to. We want new treatments and that we can use for our patients and to improve the outcome of patients. There's 3 phases in clinical trials. Phase 1 is pretty much just what dose is safe, what are the side effects of that drug. Yes, we want to see if it works or not, but that's actually not specifically the goal of the trial. Phase 2 is usually where you're looking at how effective it is. Does this work? Does this kill this specific cancer? And then phase 3 is where you actually try it on a broader scale. And a lot of times it's like, does this work better than our current standard of care? So MAP therapy for osteosarcoma. The current phase 3 osteosarcoma trial is MAP chemotherapy plus a drug called cabozantinib. Does that work better than MAP therapy alone? But to get there, we had to test cabozantinib, figure out the safe dose. We had to test whether cabozantinib worked against osteosarcoma. It did. And then now we're seeing if adding it up front improves outcome. Again, no shortage of trials out there, which is a good thing, I think, from all walks of life. Some of these are immunotherapy trials. Some of them are new drugs. Some of them are repurposing some drugs. And then we also have kind of the data commons is kind of, looking at, again, big amounts of data from past trials. How can those inform new trials? And MID Agents has actually supported the data commons in osteosarcoma. So looking forward to hearing from them. Some of the terms we use: overall response rate. I mean, ORR, again, we love abbreviations. That's how many people responded? Did their tumors shrink? Did their tumors go away? Was their disease stable over a certain period of time? That feeds into endpoints. Your endpoint determines a lot. Most osteosarcomas don't actually shrink because they're boned. Even if you kill it, it's going to take up the same amount of space. And so, tumor size might not be the best endpoint for osteosarcoma, but how long you're alive, how long before new sites pop up might be a better endpoint. There's a lot of discussion about that with the FDA for osteo. Adverse events, that's side effects, anything bad that happens because of the treatment is called an adverse event. Dose limiting toxicities, again, that's in those early phase. How high can we go with the dose? We usually aim for the maximum tolerated dose. If a little bit of drug is good, more must be better. We are learning that that might not necessarily be the case, but that was kind of how we got to where we are. Now we look for this recommended phase 2 dose. Again, phase 1 is the dose finding phase. What we want to know is what's the right dose that we need to bring to the second phase to see if it's effective. Maybe it's something low and slow that we can stay on for a long, long time. Maybe we do want the highest dose possible that you can tolerate. So those are all different. There are other things going on, as Christina mentioned. We also have another panel where it's the patients actually talking to the doctors, and we actually learn a lot from them. Sometimes it's actually most often it's rather humbling because you realize that we could do better in some regards, but that's always well appreciated from the doctors panel. We are working on a big project through Breakthrough Cancer with a lot of institutions and a lot of money going towards approaching it collaboratively in a new way. And then outsmarting osteosarcoma is the grant that we the grant process that we go through every year to kind of fund the most promising research we can find in osteosarcoma, and we'll be presenting this year's grant recipients. With that, I did go a little long, and I will try to answer any questions that I can. Again, there's way more information, and don't be shy, even at the conference, because these are complicated things. And even at the conference, any and all of us, we're happy to try to explain to it as the best of our abilities. And like I said, a lot of us probably don't know something that's not immediately in our wheelhouse. So don't be shy. Even John Healy might not know every single thing that's being presented, fully understand it.
Impossible.
Okay, maybe John will know everything, but I'll admit that I don't always know everything. But again, part of the whole conference is to educate the whole community. So don't be shy about asking questions, and it's okay.
Thank you so much, Dr. Trucco. It was a tall order to go through all of that in an hour, and you did it splendidly. So thank you so much. And as Dr. Trucco said, one of the great things about Factor is it's such a casual and small environment. It's a really intimate conference. Is that you'll have the opportunity to sit next to somebody at lunch and ask them about their work. And I am sure they will be more than happy to explain cytokines or TILs or whatever it is that they're working on. So another just great reason to come to Factor. Thank you so much, Dr. Trucco, for joining us on OsteoBites today and for making it better with us in so many ways. More information on this and all OsteoBites can be found on the MIB Agents YouTube channel, on our website at mibagents.org, and at your favorite podcast place. And we hope you can join us next week on OsteoBites. We are going to be joined by two scientists, Dr. Jose Granada Soler and Dr. Rachel Alavina, both from the University of Queensland School of Veterinary Science. As Dr. Trucco mentioned, doggies are super important in osteosarcoma research. They are going to be discussing novel immunotherapies for canine osteosarcoma and talking about a current trial they have for canine osteosarcoma. You can find our OsteoBites lineup for the next few months on our website. And if you have any ideas for future topics you'd like to hear about, please feel free to email us at events@mibagents.org. Thank you again, Dr. Trucco, for spending an hour with us today. You can get back to the 10 other jobs that you have. And thanks to all of you for joining us. We hope to see you back here next week on May 30th when we talk to Dr. Granado Solar and Dr. Alavina. And we hope to see you in person at Factor in Cleveland. Thank you so much, everyone. Thanks, Dr. Trucco.